So from a clinician's point of view, so what happens is that sometimes what happens is that the embryo quality that you finally get is poor, and we are then left having to explain to the patient why the embryo quality is poor. So although it's extremely difficult, but what we often tell them is that it could either be the sperm or the egg or us. If it's the sperm, then in a way it is what it is, but the best that he can do is to supplement himself with whatever is out there. If it is the egg, then again it is what it is, and the best that she can do is to supplement herself with whatever it is that is out there. And then there's us. Okay? And when I say us, there is the drugs, the regime, and the lab. So drugs, essentially the choice of the medication that we use. I mean, the reality of life is that you can have some patients that react well to certain drugs and who don't react well, or whose eggs don't react well to certain drugs, and I think that is true. There's also different dosages that we may have chosen. Sometimes the dosage may be too low, sometimes the dosage may be too high, sometimes the combination is required, sometimes the combination is not required. And then there's the short protocol and the long protocol, and sometimes, unfortunately, it's pretty much a trial and error. And I think every clinician accepts that, that sometimes it is, in some patients, pretty much a trial and error. So I think that one of the ways that we actually address this issue is by tweaking the protocol, trying something different, changing the dose, changing the drug, and hoping that somehow the outcome in terms of I would want to say the egg quality, but as you quite rightly pointed out, egg quality is very subjective, but somehow that the eggs that are produced leads to embryos that are much better.
Right.
And that's hoping that it all started off with the egg, because if it was the sperm, then basically, we're barking up the wrong tree.
Right. Yes.
Yeah.
Yes.
And that's normally how we look at it.
Sure.
And of course, I think you would probably agree that QC, whatever QC is there, makes sure that the lab is always...
From there, the next point is obviously we always look internally in the lab as well. When we see something not go well, we say, well, could it be us? Sure. It's always important to ask the same questions that a clinician asks about the treatment plan. The same with, was everything in order? Did we have other patients that were going through at the same time?
Exactly.
We look for obviously our quality control records. Everything's in control. Is embryo growth good generally for everyone else?
And I think this is also why it's good if you have a large lab, perhaps like ours, where you've got lots of patients running through, and each patient acts as a quality control for the next patient.
Yes.
Whereas if you have a small lab with just one patient coming through, then I don't think that you're able to compare and contrast.
You don't know that your system's
pick up
running
a
consistently. Yeah. We do a lot with tracking how well the system's running by looking at how the overall embryos are growing, so that really helps us to know system,
that all
quality.
systems are working.
Yes. And of course, there's a lot of different things we do in terms of quality and amongst the staff and amongst the different supplies we use. So those are all come into play.
Sure.
But usually we are able to rule just about everything out and say, okay, now we're at a point where we think everything is working fine, and it's either the sperm or it's the egg.
Sure.
And we go back to that question. And so then that brings us full circle to say, okay, we're back to this question of what do we do? We have bad embryos.
Sure.
They're not growing well. Is it the sperm or is it the egg?
Sure. Let's let